Today, our topic of discussion would be non-proteinogenic amino acids. So, as we've already discussed what amino acids really are, what their structure looks like, and what are proteinogenic amino acids and their structures in detail. If you have not watched that video yet, please go and watch. The link is in the description below. So let's jump on to non-proteinogenic amino acids. Any amino acid that is not naturally encoded in the genetic code of any organism is known as non-proteinogenic amino acids. Or to explain it simpler, as the name suggests, they are called non-proteinogenic because they are not involved in the protein synthesis, which we have explained briefly in our previous video. Whereas these arise during metabolic reactions, enzymatic modifications of proteins, or decarboxylation of alpha-amino acids. However, in biochemistry, over 140 non-proteinogenic amino acids occur naturally in proteins, whereas thousands more may occur in nature, but they are not present in the genetic code. Non-coded or non-proteinogenic amino acids are distinct from the 22 proteinogenic amino acids, which we've already discussed in our previous video in detail, are naturally encoded in the genome of organisms for the assembly of proteins. So, many non-proteinogenic amino acids are important. Now let's have a look at their types in detail. They can be divided into 3 types. Rare amino acids, post-translation protein modification, biogenic amines. Rare amino acids are the ones that arise during metabolic reactions. The ones which arise as a result of enzymatic modification of amino acid residues in peptides and proteins are referred as post-translation protein modification, whereas decarboxylation of alpha-amino acids give rise to biogenic amines. Now let's start with some important rare amino acids, how they are formed, and why they are important. There are 4 important rare amino acids: ornithine, citrulline, dopa, and selenocysteine. Now, starting with ornithine, which is an analog of lysine with a shortened side chain, whereas transfer of a carbamoyl residue to ornithine yields citrulline. Both of these amino acids are intermediates in the urea cycle. Let's have a look at this cycle of these amino acids before moving on to the other 2. Starting with the acetyl coenzyme A and glutamate. In the mitochondria, with the help of enzyme NAGs, N-acetylglutamate synthase forms N-acetylglutamate, which forms carbonyl phosphate synthetase 1 CPS1. Now with NH4 positive and HCO3 negative with CPS1 forms carbamoyl phosphate. Energy is converted from ATP to ADP and inorganic phosphate. Now aspartate comes outside of the mitochondria to the cytoplasm with the help of citrin. Aspartate and citrulline with the enzyme argininosuccinate synthase 1 forms argininosuccinate, which further with the help of argininosuccinate lyase forms 2 byproducts fumarate and arginine. Arginine with the help of enzyme ARG1 further forms 2 products, urea and ornithine, whereas ornithine is transported into mitochondria via ornithine translocase 1. Carbonyl phosphate with OTC, ornithine transcarbamylase, forms citrulline from ornithine. This is a continuous reaction during carbonyl phosphate synthetase. Now moving towards the other 2 rare amino acids. Third rare amino acid is dopa, which is an acronym for 3,4-dihydroxyphenylalanine and is synthesized by hydroxylation of tyrosine. It is an intermediate in the biosynthesis of catecholamines and of melanin. As you all should know, the catecholamines are biogenic amines that have a catechol group, and melanin is a natural skin pigment. Levodopa is a drug made from dopa. Levodopa is an excellent replacement of dopamine, used in the treatment of Parkinson's disease. Now coming towards the 4th rare amino acid, which is selenocysteine. It is a cysteine analog. It also occurs as a component of a few proteins, for example, in enzyme glutathione peroxidase, which is a seleno enzyme and can be called as seleno-glutathione peroxidase. It has a peroxidase activity whose main biological role is to protect the organism from oxidative damage. Now coming towards the second type, that is post-translational protein modification, which is basically defined as the chemical changes that occur after a protein has been produced. It can impact the structure, electrophilicity, and interactions of proteins. Electrophilicity is the property of being electrophilic, that is, having an affinity for electrons or electron-loving. Now let's talk about their types. The post-translational protein modification is based on the addition of chemical groups, addition of complex groups, addition of polypeptides, cleavage of proteins, and on the amino acid modification. Now let's talk about the post-translation modification based on the addition of chemical groups. There are 4 types of chemical groups: phosphorylation, acetylation, hydroxylation, and methylation. Starting with the addition of the first chemical group, phosphorylation. So, addition of a phosphate group on serine, threonine, or tyrosine residues, which forms phosphosyrine, phosphothreonine, phosphatyrosine, is known as phosphorylation of proteins. Phosphorylation is performed by enzymes called kinases. Phosphorylation has implications in several cellular processes, including cell cycle, growth, apoptosis, and signal transduction pathways. The second one is acetylation. Acetylation refers to addition of acetyl group in a protein. For example, acetylation of lysine will form acetyl lysine by the enzyme KAT, lysine acetyl transferase. It is involved in several biological functions, including protein stability, location, synthesis, apoptosis, cancer, DNA stability. The third one is hydroxylation. This process adds a hydroxyl group O to the proteins. It is catalyzed by enzymes termed as hydroxylases and aids in converting hydrophobic compounds into hydrophilic compounds. And now the fourth one, which is methylation. Methylation refers to addition of a methyl group to lysine or arginine residue of a protein by enzymes called methyltransferases. Now coming towards the post-translation modification based on the addition of complex groups. There are 3 types of complex groups: glycosylation, amylation, lipidation. Glycosylation involves addition of an oligosaccharide termed glycan to either a nitrogen atom, which is called as N-linked glycosylation, or an oxygen atom, which is called as O-linked glycosylation. N-linked glycosylation occurs in the amide nitrogen of asparagine, while the O-linked glycosylation occurs on the oxygen atom of serine or threonine. They have critical roles in protein sorting, immune recognition, receptor binding, inflammation, and pathogenicity. Now the second one is amylation. Amylation refers to addition of AMP, which is abbreviation of adenosine monophosphate, to a protein. It involves formation of a phosphodiester bond between the hydroxyl group of the protein and the phosphate group of AMP. And now the last one, lipidation, which is basically the covalent binding of a lipid group to a protein. Now coming towards the post-translation modification based on the addition of polypeptides. It have ubiquitination. Ubiquitination involves addition of a protein found ubiquitously termed ubiquitin to the lysine residue of a substrate. Addition can be of either a single ubiquitin molecule, which is called as monoubiquitination, or a chain of several ubiquitin molecules may be attached, which is called as polyubiquitination. Now coming towards the post-translation modification based on the cleavage of protein. It is consisted of proteolysis. Proteolysis refers to breakdown of proteins into smaller polypeptides or amino acids. Now coming towards the last post-translation modification, which is based on the amino acid modification, which consists of deamidation. Deamidation is the removal or conversion of asparagine or glutamine residue to another functional group, such as asparagine is converted to aspartic acid or isoaspartic acid, while glutamine is converted to glutamic acid or pyroglutamic acid. This modification can change the protein structure, stability, and function. Now coming towards the last type of non-proteinogenic amino acids that are biogenic amines. Several amino acids are broken down by decarboxylation. This reaction gives rise to what are known as biogenic amines, which have various functions. Some of them are components of biomolecules. Let's have a look. The amines, ethanolamine, cysteamine, beta-alanine are derived from amino acids, serine, cysteine, and aspartate respectively, and functions as the component of coenzyme A. Biogenic amine. Aminopropanol is derived from threonine, which functions as component of vitamin B12 gamma aminobutyrate, is derived from glutamate, and is an important neurotransmitter. Histamine is derived from histidine and functions as a mediator and neurotransmitter. Dopamine is derived from dopa, which also functions as a neurotransmitter. Serotonin is synthesized from tryptophan, functions as mediator and neurotransmitter. Okay, so that was it with the non-proteinogenic amino acids. Let's have an overview. Non-proteinogenic amino acids are divided into three types: rare amino acids, PTM or post-translation modification, and biogenic amines. Important rare amino acids are ornithine, citrulline, dopa, and selenocysteine. PTM are based on the addition of chemical groups, addition of complex groups, addition of polypeptides, cleavage of proteins, and on the amino acid modification. Further PTM, which are based on the chemical groups, have phosphorylation, acetylation, hydroxylation, methylation. Complex groups are comprised of glycosylation, amylation, lipidation. Ubiquitination falls in the addition of polypeptides. Proteolysis falls in the cleavage of proteins. Demidation falls in amino acid modification. Now, the third type of proteinogenic amino acids, which is biogenic amines, are ethanolamine, cysteamine, aminopropanol, beta-alanine, gamma-aminobutyrate, histamine, dopamine, serotonin. That was all about non-proteinogenic amino acids. Follow sqadia.com for more biochemistry videos.